So, what we have here is a system without the inducer. They have they put this F prime segment in here. They can follow whether this is being expressed by looking at the mutated form of permease, or they can see it being expressed up here for this operon by looking at the mutated form of the beta-galactosidase. So without the inducer, this was turned off, but they could pick up this one because they modified this operator so it wouldn't bind the repressor. So they always got this being expressed. So this literally became constitutive in its expression. Now, when they add the inducer to this system in this bacterial cell, now suddenly they have knocked the repressor off of this, and now they get expression of this particular mutated form of beta-galactosidase. So that was one of the experiments. There were multiple combinations of this in play. They were doing all kinds of things to come up with this. All right. Now, if they have a mutation in the actual repressor gene to where this cannot bind to the operator region, now they have constitutive expression. If they add this system, have this in the F prime segment, and add this to one where the E. coli chromosome segment, what they found was with a normal repressor gene, it would not only bind to this to turn that off with that one, it would turn this one off that had this one. As a result of that, they could see how the repressor system works. With the inducer, when they bound this to have these two in here, then they found that they got gene expression here and here. So they just went through a series of these things, just different ways of doing this. Okay? They also did, that was sort of a recessive type trait because that gene is not functional, but they found another one which was a dominant trait, meaning that here's the normal lac repressor, but here's a mutated form. But this mutated form lacks the ability to bind allolactose, but it still retains the ability to bind to the operator. As a result, if you're expressing these and you're expressing this, if it binds over here and binds over here, no matter if you add allolactose, this prevents the gene expression. So it's a dominant impact on this gene expression system. If we look at the actual polypeptide, the polypeptide is that's associated with the repressor. It actually is 4 of these are brought together. The DNA binding domain are these helix-turn-helix structures up here. If we flip this over and bind to a DNA operator region, so here you can see this binding to the DNA, and you can see the helix-turn-helix, which is interacting with a major group. And in fact, if you show this more properly, this is actually looping out the DNA like this, and preventing and blocking the ability of the RNA polymerase to get past this. So the RNA polymerase is either on this side, it's trying to get past this, but it's blocked this way, and so it can't get past of it. All right. So this data that was presented by Jacob Monod pretty much convinced most scientists at the time that all genes were under negative control due to the binding of repressor. Well, that's kind of a big jump. That always kind of happens in science, but what we've learned in science is to never say never. You cannot say that it's only this. There's always a possibility because life has a tendency to change things, okay? So keep that in mind. Now, about this time, so one of the other things that they actually discovered when they were studying this, they showed that it was under an inducible control and a negative control. Okay, those were the two main ones they looked at. But they also, when they were studying this, they also found that there was a positive control mechanism in place, so that once the lac repressor was removed and you're having lactose expression, there needed to be something in there that would promote a faster expression of those particular genes, because you have a ton of lactose suddenly in the environment. So there was a positive control system on the lac operon, where it turns on the expression of the lac operon. So once you remove the repressor, so the repressor is moved off the operator, there is a protein called CAP. And CAP is catabolite activator protein. When glucose is not present, there's a higher level of a molecule called cyclic AMP. It's a signal molecule. And it will bind to this protein, change its shape, and make it active. Okay? That has to be in the absence of glucose. When that happens, now this is set up and it promotes the RNA polymerase and its sigma factor to come into play, bind, and then take off and do gene expression. It actually brings them in faster. So that, because it needs to have these genes turned on at a higher level to utilize the allolactose. Now, what we're going to see happening in the next few slides is we're going to see a situation to where we deactivate this whole system. And the mechanism for deactivating is just adding glucose. Okay? So, the role of lac repressor, this is turned off. The cyclic AMP is bound to this. You got it to the cap. You're really taking off with a higher rate of transcription. Okay, so that's with this. If the repressor is present, it binds. Transcription is going to be very low due to the binding of this. Okay, but this guy is still in place. All right. And you eliminate this, even with the cap off of it, there's still some transcription because you've eliminated this. If this is not bound and this is bound, there's very low due to the fact that that's blocking it. All right, so that's a review of it. So what happens when you add glucose? One of the first things that happens is you're going to remove that cap protein off of the actual promoter region of the lac operon. And what happens is that glucose will inactivate the enzyme that generates cyclic AMP from AMP. So AMP itself, an enzyme called adenylate cyclase, creates a cyclic structure. Here's the phosphate that creates the new cycle that calls the cyclic AMP. What it does, it eliminates two phosphates off here and links that phosphate to that hydroxyl group at the 3' portion of the carbon. And that becomes a signal molecule. It's an ancient signal molecule used by multiple organisms and during evolution. We still use it as well. Now, if this is present, suddenly you throw glucose in. Well, glucose will then inactivate this by using another gene to turn this off. It will also activate an enzyme that will destroy all cyclic AMP within the cell. So it eliminates it and no longer has a cycle. Now you don't have that. Cyclic AMP is necessary to bind to the cap protein. If it's not present, what will happen now is the cap protein complex will come off. So that's the initial stages of turning off the whole system. Lactose is no longer present, so you can't generate any more allolactose. The repressor starts to bind to it, and then what you do is you basically turn off the system. So because of the presence of that, it reduces the amount of that. We have it set up. So if we look at this more in the detail of the genes themselves, you can just see how this thing fits. So here is the start site for the gene expression in this gene, and it actually has a GUG start site for, in this case, for methionine. But if we look at the base pairs and how they line up, you can see how this whole thing is interacting. So here's the cap binding site, right here for the cap site. Here's the RNA polymerase binding site. But note here is the operator, and that's the part that's bound to the repressor. So there's binding sites, and it binds this and blocks this. Note here that this is the start site of transcription right there. So that repressor is sitting right on top of all of that. But the RNA polymerase can be sitting here. It's just waiting for this guy to come off so it can start transcription. And you can see how the whole thing comes together here for this whole system. You even have a Shine-Dalgarno sequence, which is down here in front of where the start site of translation begins for that enzyme. So it's quite interesting how the system is set up. All right. So that was the initial model. And at the same time, there was other individuals who were not just looking at utilization of lactose, which is breaking down a compound. They were looking at the production of structures necessary for polypeptides. For example, they were looking at the genes necessary. And what they found was there was one called the tryptophan operon. So just like in the utilization of lactose in the tryptophan operon, there are structural genes here that are necessary to generate enough tryptophan for the system to work. So there are 5 structural genes in this, and it's just like before, it's a polycistronic system where you generate all 5 of those genes necessary for that biochemical pathway necessary to produce tryptophan. Okay? So all necessary amino acids may not be present in a growth media, which means that growth media, they're going to have to take some things in there and put it into a biochemical pathway to generate those amino acids. If a specific amino acid is missing, the bacteria has certain operons that enable the bacterial cell to manufacture that amino acid. So this is the tryptophan operon that is set up. Just like the other one, it has a promoter, an operator, but built into this is something else. It's called an attenuator system, okay, or leader region. This is a small area that on the messenger RNA that's going to come off here that is going to bind the ribosome initially. And in this area are multiple trip codons. And what happens here is this becomes what we refer to as an attenuation gene expression, an attenuated gene expression, meaning that in the presence or absence of tryptophan, there's going to be an influence on how many of these structural genes you produce. Okay? That's where this comes into play. So, at low tryptophan levels, that means you need to make some tryptophan. What will happen is you will activate gene expression. Now, like before, you have a negative repressor to the system. It's bound to it and it's going to prevent the expression of this system. Okay? Now, this negative repressor does a little bit different. It has an effector molecule that binds to it. And in this case, the effector molecule is actually tryptophan. If there is a high level, if there's tryptophan in the environment, you don't need to have this genezyme. You don't need to turn on these structural genes. Thus, it acts as an effector molecule. What it does is you have a repressor that is being constitutively expressed. If tryptophan is available, it binds to this, changes its conformation, and it goes in and it binds to the operator region in front of these tryptophan structural or tryptophan operons and blocks any gene expression of these because it doesn't need it. Okay, so it's a little bit different than the other one, but in this case it represses gene expression. So high tryptophan levels, repression occurs. So there are two mechanisms that are playing a role here. The first one is the repressor-operator interaction, okay, with tryptophan. So you have a particular system set up to where you have an operator region right here in front of the promoter. With tryptophan as the effector molecule, it binds an aporepressor that binds to this and blocks any gene expression. It turns off the gene. If the tryptophan is not available, the repressor can't bind, and you get this long polycistronic messenger RNA. Note here, there is a region of it called the leader that came off here, which I'm going to talk about here in a moment. This is actually used as a volume control on how many of these genes are expressed. So if you can think about it, in each cell there has to be, based upon what polypeptides it's producing, it has to have a certain amount of tryptophan. But it doesn't have to have a high dose of tryptophan sometimes. Sometimes it just needs a low dose of tryptophan to keep everything operational. Okay? If there's too little tryptophan, then the system starts to slow down. So there has to be a mechanism in place to kind of activate it up and get it to a level to where it's supposed to work. But at the same time, if it gets too high, that's a waste of energy. So there has to be a feedback system to this that says, okay, we'll turn down the volume. That's what this whole thing is about. And this is the next level of mechanism of regulation. It's called attenuation control regulatory leader, and it involves this that is being expressed here. It's going to be this area that's going to lead to this system. The thing to remember about bacteria is that a bacterial cell, when you're generating a messenger RNA, at the same time, the ribosomes can bind. As soon as this comes out far enough, the ribosomes can start binding and generating the polypeptide. It doesn't have to go outside of a nuclear envelope. There is no nuclear envelope. You literally can have the ribosomes binding to this thing and taken off. So attenuation control involves this leader, determines if initiated transcripts include other structural genes or not. If there's too much tryptophan, then this will block these other genes being activated. So as I was talking about this particular system of control, this is called the attenuation regulatory leader region. And in this region, this is playing the role of regulating the translation of this particular polycistronic messenger RNA. I also want to point out here, this is demonstrating where each one of these enzymes are participating in the biochemical pathway in the production of the L-tryptophan. So you can see how they can play a role in this whole system. and it allows us to be able to actually assess which ones of the enzymes are working. So let's kind of get started on this. So first of all, the operon is called a repressible operon. So there's no transcripts in presence of tryptophan. If tryptophan is available in good quantities, the system's turned off. It's not needed. So basically the biosynthetic pathway is catalyzed by these specific enzymes at each step, which is coded by the specific genes. And so they're going to keep these genes turned off if if tryptophan is present at a good enough quantity. All right, so in the attenuation control, we have a situation, absence of tryptophan or in the presence of low amount of tryptophan. This is going to be the volume control here. This is good. If there's not enough tryptophan, this system's going to turn on to allow more tryptophan to be produced. If there is enough tryptophan, then the first method of repression, where the tryptophan binds to the repressor, it binds to it and blocks the whole thing, that will be the major point of repression. But in this situation, there's just not enough tryptophan, and it's going to allow this system to turn on. So, under very severe tryptophan starvation, gene activity is at maximum. That means that we get the full, all 5 structural genes produced. So that's why we refer to those as the long transcripts. Huge long transcripts are produced. However, under less severe situation, gene expression is less than maximum. In this particular case, that means that it's just a little bit down, and it just needs to have a few more structural genes expressing themselves. Then what will happen is you'll have some long transcripts, but because of that leader region will actually cause the RNA polymerase to pop off, and you'll end up with short polycistronic messenger RNAs, which only have a few of the structural genes. And that's the concept of the short transcript. So, greater the amount of tryptophan, the greater the number of short transcripts, to the point to where it finally just cuts down the shuts down the gene expression using just the tryptophan repressor system. So attenuation control terminates transcription, producing short transcripts. So here's the messenger RNA. So if we look at the messenger RNA here, this is how it's organized in the leader region. So what we're looking at right here actually is this little leader. We're just looking at that very short, we're not even looking at any of these except for the start site for that particular one. So this is the leader. And if you'll notice, this leader has a start site codon. But there's an interesting codons in here. And if you look carefully in this leader region, look here. This is the leader peptide codon region. There's some tryptophan codons there. Well, let's think about that. Tryptophan codons. That means that this is where the transfer RNAs that have tryptophan linked to them are going to come in to allow for a very short peptide to form. But if you don't have any tryptophan codons, then whatever the ribosome here is coming along, and it comes along here to where you don't have the, not tryptophan codons, but tryptophan charged transfer RNAs, then the actual ribosome is going to stop here at this region we call region 1 of this leader. It just stops there and it sits on there because it's waiting for a transfer RNA. It's being blocked. But if you do have the tryptophan charged transfer RNAs, it'll go past here, it'll go all the way down here, and you'll have the ribosome sitting on top of this number 2 region. Well, in that leader area, there's some interesting structural elements here. We call them regions 1, 2, 3, and 4. When this transcript comes off, it can sometimes form into a hairpin loop. So for example, region number 1 and region number 2 can form hydrogen bonds with each other. Region number 2 and region number 3 can also form hydrogen bonds, and region number 3 and region number 4 can also form hydrogen bonds with each other. So you can have some of this three-dimensional shape created in there. So if there is no ribosome attached to it, these things can form. The question is, which one forms? Well, let's talk about that. Transcription and translation is going to be tightly coupled in these particular prokaryotes. Attenuation occurs at the messenger RNA level and can reduce transcription of the trip operon 8 to 10-fold. So, what happens as that messenger RNA is coming off and that leader comes off, there's going to be a binding of 1 to 2. It's called the pause of translation. So what happens here is that this structure will form, and it basically pauses any type of translation long enough for the ribosome to load onto the messenger RNA. So 1 and 2 come together, and now the ribosome loads on and they open up. The position of the ribosome on the leader transcript determines if transcription is terminated or not. That's the goal here. So the ribosome is now coming in here, all right? If no tryptophan transfer RNA, then the ribosome will stall at this point. It'll stop right here. Just sit right there on top of it. Now, region number 1 can't bind to region number 2. So what will happen is region number 2 will interact with region number 3 as it's coming off the RNA polymerase. So the RNA polymerase is right over here, and the ribosome is already attached and moving along. And suddenly this region number 2 to number 3 comes together. Well, because of that it's far enough away from that RNA polymerase, we refer to this as the anti-termination, meaning that this structure, if this happens when there's no tryptophan transfer RNAs to bind here, causing the ribosome to stall here, when this structure forms in the actual RNA molecule, then that prevents any type of termination of transcription. The RNA polymerase will keep going. Alright, so that, if that happens, so the 2 and 3 loop for anti-termination forms, thus region 3 can't pair with region 4. If region 3 can't pair with region 4, we have anti-termination. But here's the interesting part of this. If region 3, if this goes past this and comes, the ribosome ends up here, then region 3 and 4 are now able to combine. That's close enough to the RNA polymerase as it's being used that if this forms, it will actually cause the RNA polymerase to pop off of the gene, and we'll get a short transcript. That's enough of a three-dimensional structure to cause it to come off, and now we have a short transcript, all because we had some tryptophan charged transfer RNAs over here. So we'll review that again. As the RNA polymerase is going along here, the ribosome attaches. It starts to move along here, making a very short peptide, and as it gets to these tryptophan codons, it's going to look for tryptophan charged transfer RNAs. If there are none there, it'll stop and it will kind of hold up here. All right? As a result of that, that prevents region number 1 of this leader messenger RNA from binding to region number 2. That frees up region number 2 to bind to region number 3, and this creates a three-dimensional structure in the RNA which does not interfere with the RNA polymerase continuing its transcription. However, if you have tryptophan charged transfer RNAs, which means you have tryptophan, a lot of tryptophan in the system, then what that does, it goes past that, ends up on the region number 2, which prevents it from interacting with region number 3. And if region number 3 can't interact with region number 4, if region number 3 can't interact with region number 2 because the ribosome is sitting here, then 3 and 4 interact, and they're close enough to the RNA polymerase to cause it to pop off, and you get a short transcript. Now, depending on the amount of transfer RNAs in there, if you have a good amount of transfer RNAs, tryptophan, then that tryptophan is going to keep all the transfer RNAs charged, and so pretty much the system doesn't need any more tryptophan generated. But if you have like maybe 50% of normal, then yeah, you're going to end up having long transcripts formed every once in a while and short transcripts formed. So there's the volume control in this system. And you can see here how it works. Here's basically region number 1, right here, interacting with region number 2, which can interact with region number 3, which can interact with region number 4. This is just showing how these all can actually interact with each other. As I mentioned, as the ribosome is coming along, it pulls this off, and if it pulls that off, then region number 1 doesn't interact with region 2, and so now you have this terminator loop. It comes along here and blocks this, so now you have the anti-termination. If it goes past that and continues down to here, then the ribosome is sitting on this region number 1 and 2, then basically it interferes with this and this guy forms. And if that guy forms, then you're going to knock the RNA polymerase off. So here's a way of looking at it. So here, this is the anti-termination. It comes along, it stops on top of the codon for tryptophan. Now this interacts with this one. So that's the anti-termination group. If it continues, then this portion becomes the predominant one because this moves over, and now that interferes with the RNA polymerase and knocks it off. And so you have a termination signal for transcription. Thus you get a short transcript. So here, star for tryptophan, transcription continues. Plenty of tryptophan, and you terminate the amount. Thus you don't waste the energy making those particular proteins that are not needed. This attenuation control is used in several different types of bacterial operons for generating amino acids. Here's some for phenylalanine, histidine, leucine. These are the leaders that have been there. And some of these are like isoleucine and threonine can kind of in combination with each other. So this is a very common system for all the different amino acids. All right. So what that introduced us to is that with the lac operon, it created a concept that all genes are under negative control. And then there was sometimes built-in attenuation control, volume control on these. So that introduced us to that concept. About the same time, another group was looking at another sugar and how it's being utilized by bacteria. It's called arabinose. They pointed out that in their particular system, that instead of being primarily negative control, their system was turned on by a positive control system, which was contrary to everything that was being pointed out by Jacob and Minot and some of the other scientists. So it wasn't well taken. It wasn't well received, but over time it's been well received. So it's called the arabinose or ARA operon. Positive and negative control. It has a negative control also, but for gene expression, it's actually the positive activation that brings about gene expression. But it's got an interesting system to it. So let's take a look at this. At the same time, as I said, they were talking about it, and they found that instead of being regulated with negative control mechanism, it is a lac operon. Although their conclusions were not widely accepted, biochemical and molecular tests proved it to be. So if glucose is present, there is no cyclic AMP. CAP comes into play here as well. And so you have no gene transcription if glucose is available. So if we look at this closely, just like in the other ones, you have an operator system. You have, in this case, their inducer sites are called arrow I1 and arrow I2. And it has the promoter and things like that. Plus you have polycistronic messenger RNAs for this whole thing to be utilized. It also has a cap site back here. Now, this inducer sites, we call them inducer sites because these things are going to be binding not only to the repressor structure, but they're also going to be binding to the inducer structure. And I'll show you what's going on here. Now, there's an operator up here. This is basically part of this system here. What's necessary is this creates a bending loop. So if you'll see here, here's the operator system up here for this ARA C here. ARA C is this protein right here that's being produced. When it's produced, it binds to ARA I1 there, and it's a dimer, and it creates a bridging between that to the ARA O2. I'll show you a picture here in a moment. That creates a negative control. So if there's no arabinose around, this is the structure that it happens. But here's the interesting part. So it is it's negatively deactivated, but here's the part that causes positive control. When arabinose becomes available, instead of knocking off these negative regulator proteins, what it does, it converts them into positive regular proteins. So the arabinose binds and it causes this to flip down, and now the dimer system's set up to where you're binding both of these inducer sites. That has to be in place. It has to be in place to attract the RNA polymerase. So you can see this is a positive activation of the gene, not a removal of a negative control. What it's doing is the system's already set there, and it's just waiting for the effector molecule, arabinose, to come along and it comes along and it modifies its shape into a positive regulator. So now it brings in the RNA polymerase, because that's when the RNA polymerase comes into place. So now you have a positive activation, an inducible activation of this particular gene. To keep it on, the CAP and cyclic AMP are in place, and then once if glucose becomes available, then that will help to deregulate and turn this whole system back off. So you have this, for the arrow operator to function, glucose cannot be present. If present, it will eliminate cyclic AMP and focus on the utilization of glucose. So for arrow C to bind to arrow I2, its conformation has to be modified by arabinose. All right. Now that arrow C shape is modified, it binds to the arrow I2, okay, which is needed for RNA polymerase to come into it so that you can get the promoter to be activated. So thus the gene expression is a positive induction of gene expression. It's not a removal of a negative factor, it's an induction of gene expression, and that's where there was a contradiction to what Jacob and Minot were talking about. So positive regulation of activators is now known to occur in a variety of prokaryotic systems and in all eukaryotes. All right. So, inducible versus repressible regulation. How does this play a role in a variety of systems? They studied a lot of operons, and they revealed a general trend concerning inducible versus repressible regulation. Operons in the bacteria involved in catabolism, meaning the breakdown of a substance, are typically inducible. The substance to be broken down or a related compound act as the inducer. Okay? Operons involved in anabolism, that's biosynthesis of a system, are typically repressible. The inhibitor or co-repressor is the small molecule that the product of the operon. So this would be in the case of the lac operon or the era operon, and this would be in the case of the tryptophan operon in that particular system. So it's a really nice, interesting thing to kind of keep in mind when you're looking at these particular systems. Now, there's a series of videos that are about to follow that I would like for you to watch on your own. These are actually leading to eukaryotic gene expression and how this plays a role in shaping our life. This first one is the shape of life, and it's talking about these genes called Hox genes. Hox genes have been found to be at the foundation of the organization of all eukaryotic organisms, where it creates special organisms that have segmentation in their particular structural build. And so these Hox genes have been utilized all through evolution in that particular system. So I would like for you to watch that and give you a little bit of a format to understanding why we're talking about these systems. In addition, eukaryotic gene expression, these are really, really good discussions about eukaryotic gene expression. These are good videos, and I would like for you to watch these as well to help you understand in the next system that we're going into. Any type of video, these two actually have really nice animations that are showing things moving around, which gives you a little more information as to how things work, okay? So make sure that you watch these. And then what's interesting is seeing how a limb can be regenerated and how our understanding how gene expression is occurring in eukaryotes gives us a lot of information about how these use these genes to be able to create structures of our body. So this ends our particular lecture on this particular chapter, and we'll be starting in chapter 15 on the next lecture. Thank you, and I'll talk to you soon.